So we are ready to treat our cells. And today is day 2, so this is 24-hour time point. And we are going to start off by going ahead and labeling our plate so we know exactly how we are going to treat them. So I'm going to have our untreated, MS023, control, MG132, and then our drug at low, medium, and high concentration. And we're going to treat them exactly the same way as we treated the ones at 48 hours, and we'll treat them the same for the other time points tomorrow. Now, remember, for treatment, our media is 2.5% containing FPS instead of 10%. We just simply diluted it down with some base media, the base del bucco modified Eagles medium, to bring the concentration down to 2.5%. And we're going to wash our cells with some phosphate buffered saline. This is just physiological saline to clean up the cells and remove remnant proteins and dead cells from the cell culture before you treat them. And then, of course, we have our drugs. We decontaminated everything with ethanol right before we used it, and now we are ready to use them. The first thing we're going to do is switch on our aspirator, and we are going to clean it with conflict. We've already looked at the cells. Cells looked healthy and happy at about 50 to 60% confluent, depending on the well. If you're ever unsure if you touch the incorrect space outside the culture, always decontaminate again, just like I did. So we remove the media. The next step is to wash the cells with PBS. So we will take our serological pipette, gonna take PBS, and we're going to wash them with about 2 ml per well of PBS. It's important to be gentle and quick so you remain aseptic and don't disturb the cells. Not so important to be exactly 2 ml. You don't want to do more than 3 ml, but you just want sufficient volume to cover the cells. Now, if you have to prepare treatments or do anything else, you should have done it already. Like I've made the media already, so that you don't have to wait once you remove the wash buffer, because the cells get stressed if you leave them without a liquid. Now I'm just removing all the wash buffer. There's some media left over here. I'll collect that too. And then I'm gonna decontaminate again. And now I'm ready to switch them to serum-free media, or in our case, 2.5% containing treatment media. So to begin with, I'm just going to take 2 mLs in each well. This time I do have to be exact because our final concentration of drug depends on that. So I'm just going to go carefully to the edge and of each well along the wall and put in 2 ml of media to each well. If you have a bubble, take it out. That's it. And the last step is to treat the cells with the drugs. So our first treatment is our MS023, and we need 2 microliters in 2 ml to get the concentration we need. So this is a 0.2 to 2 microliter pipette. So this is my MS023 that I'm treating the cells with. So there we go. And I'm treating them with, I believe, 100 micromolar. Then for the next one is my MG132, and we need 1 microliter in 2 ml. And this is a 50 millimolar stock, and I need 25 micromolar final concentration. So I'm going to get it accordingly. We just have a little bit left, so it's in here. Otherwise, if you have a lot of amount, you want to always aliquot. And then for the last one, or for the last samples, which are our P2, low, medium, and high. For the lowest concentration, we need 20 microliters in 2 ml, which will give us 10 micromolar final concentration. Our stock is 1 millimolar P2. We can switch off the aspirator actually, since we don't need it. So for P2, I have 3 wells. The first one, the low one, gets 20 microliters for a final concentration of 10 micromolar. That's our working concentration of the drug. For our medium concentration, we're going to use 40 microliters of 1 millimolar P2 and 2 ml, and that's going to give us 20 micromolar of the drug. And for our final pH, so the highest concentration, we are going to be treating with 40 micromolar, and for that one we are using 80 microliter of the drug. Because we were using the same drug in all the wells and I never actually touched the media, we could use the same pipette tip. And that's it. We are ready to put them back in the incubator and clean up.